Okay, so we are at Bio 212. This is going to be part 3 of chapter 26. I'm probably going to be part 4 as well. And we're in the continuation of the materials that have been filtered into the glomerular capsule of the nephron, and we're now working our way through the system. So I'm going to draw that out for you one more time. But before we get going, I don't think I said this is Norwalk Community College, but it is. And here's what I'm going to do. We're going to go like this. I'm going to do something like this. Here is the... It goes like that. Proximal convoluted tubule, descending limb, ascending limb. Notice this time I'm drawing the difference. Distal convoluted tubule and collecting duct. And now in red, I'm going to make sure you see this piece here. And that's the afferent arteriole that's coming in, creating our capillary bed with our podocytes, our mesangial cells. And notice now what I'm doing is I'm making sure that you see that that's where that macula densa is. Okay? So now everything that's been filtered, we're now in the chemical composition of tubular filtrate. Okay, it's quite different from filtrate. Because remember, at our proximal convoluted tubule, we reabsorbed, let me change colors here so I don't get us confused. We reabsorbed as much as we could out of the material right as it moved in. Okay, so I'll put that over there, draw this up here like that, come around, ting, 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 and this ultimately will give us urine. Right? So, inside this space here, this proximal convoluted tubule, we reabsorbed as much as we could. And now we're moving into the lower end of the loop of Henle, and then we're going to ascend. So by the time we actually get from, let's say, here to here, where's all that material gone? Well, it's gone into this space here, where now you actually have, let's go back to red. Let's put a capillary bed inside of here for you so you can actually get the general idea. Okay? And then we'll put some blue there. And because this is a capillary bed and it's very thin, you can now collect all of this material and send it out of the actual kidney itself. Okay? Does that make sense, I hope? Okay, let me make sure you go back to red here. So you make sure this is the afferent arteriole here. There's your efferent arteriole, which we're going to make up our capillary bed from. We're going to collect all the waste materials. For the first time, reabsorption of water is not automatically coupled with reabsorption of solutes, okay? Where you're going to actually have the potassium, sodium, chloride symporters that function in sodium and chloride reabsorption, okay? And there's little or no, this is the important part here. Let's get rid of the drawing. Let's go like this. There's little or no water reabsorption in the ascending limb. Okay, and that means the osmolarity inside of the tube is decreasing. But that also gives us a gradient. That's what's important here. Okay, there's no reabsorption. Okay, here's the example. Here's what they're talking about. Notice they've switched their cartoon around so it fits their artistic needs, but this isn't what it would actually look like. Because remember, the macula densa has to be over here. So, sodium, chloride, potassium. Potassium kicks back in, depending on what we need. They can pass here through pericellular transport, but for the most part, chloride goes out, sodium goes out, and what we're doing now is we are regulating the osmolarity. And notice what they're doing as well. They're telling you exactly where this is happening. So this is only happening in those juxtamedullary nephrons because the blood vessel here is the vasa recta. Okay, that's how we know that this is the only place that is happening. Okay. How do we actually now begin to regulate this? Because the late distal, okay, convoluted tubule and collecting duct, where reabsorption on early distal convoluted tubule will be sodium and chloride. This is the major site where parathyroid hormone stimulates calcium reabsorption. Okay, and remember I told you about the principal cells and the intercalated cells. I'm just going to underline this because if I make it bold, it's going to just completely blow off the page. Okay, so principal cells reabsorb sodium and they secrete potassium. Intercalated cells reabsorb potassium and bicarbonate so that you can secrete hydrogen protons. That's what we mean by regulating pH inside the body. Okay, cool. So how do we regulate it? Well, fine well what we're going to do. We're going to use angiotensin 2, bold. We're going to use aldosterone, bold. And notice we're talking about calcium here. I mean, we didn't care about calcium before, right? When we were talking about regulating blood volume or water and all that sort of stuff. But we don't really care. Mother Nature doesn't really care about that. She needs the electrolytes that are required for normal bodily function inside of you, right? So here are our major hormones. Angiotensin 2 decreases glomerular filtration rate. It enhances reabsorption of water, of sodium and chloride as a result of water in the proximal convoluted tubule. Okay. Aldosterone, when blood volume and blood pressure decrease, stimulates principal cells into collecting, okay, to reabsorb more sodium and chloride and therefore secrete more potassium. This keeps you alive, right? It means your heart rate doesn't get all wonky on you. Right? And then parathyroid hormone to collect, reabsorb more calcium, because we all know we need that muscle function, nervous system function, stuff like that. Okay, so antidiuretic hormone increases permeability. Notice now we're telling you exactly where it's working. It's going to insert a different protein. That different protein is aquaporin 2, not aquaporin 1 from the previous slide. Atrial natriuretic peptide. There are two sources for this. We only talk about the one inside the heart. There's also brain-derived natriuretic peptide. Okay. What we're going to do, of course, is notice what it does. It suppresses the secretion of ADH and aldosterone. Okay. And what it's going to do is it's going to decrease blood volume by inhibiting the reabsorption of sodium and water in the proximal convoluted tubule. That doesn't seem right. It should be distal convoluted tubule. There's distal. No, we'll say proximal. Okay, because that's going to get the larger volume in. Correct. Okay, proximal convoluted tubule. But I'm going to go double check that. Okay, cool. Everybody with me? Cool. So now, essentially, if you went back to those last three slides, right, you're going to see that I'm starting to talk about which ions are being moved by which hormones. What's the consequence of each? And what we really want to do is we want to figure out why Mother Nature is doing this. And that's because sometimes she either needs to void dilute urine or make sure she's voiding concentrated urine. So you can tell when this is going to happen. You have too much water inside your body or you have too little water inside your body. ADH controls whether dilute or concentrated urine is formed. ADH, antidiuretic hormone. If it's absent or low, you're going to have dilute urine. If you have high levels of ADH, you're going to have more concentrated urine. And that's because, remember, ADH targeted specific sets of cells to control the flow of sodium ions. Okay. Now things are going to get weird. And by weird, what I mean is we're going to focus our attention on the juxtamedullary nephrons. Okay. Where we need to think about what the osmolarity is of the fluids inside of the tubes. What you're looking at here, aside from getting really weirded out by all the numbers, is you have essentially even osmolarity as materials enter into the proximal convoluted tubule, right? And this is the medulla, and you're going to see that it stays pretty much constant until we get to the loop of Henle. Then remember, on the ascending limb, it becomes impermeable to water, which means we're going to change the osmolarity. Okay, and you're going to see that we've restricted the osmolarity as we make more and more dilute urine in this case. Okay, so we're allowing the extra fluid inside of our body to not absorb water to not absorb water, moving in the opposite direction. Here, let me put an arrow inside there. If we wanted to keep that water, at this point in time, we would send water out here, right? And the values would become more close to one another. But because we want it to be dilute urine, we allow this to become essentially dilute urine. And what I mean by that is specifically, let's look at the osmolarity. It's 65 versus 300. So this is 5 times more dilute, if you will. Okay. I should check the math on that. But hopefully you get the idea. If it's most concentrated here, it's 900 milliosmoles. Here it's 65 milliosmoles, and that's why you end up with dilute urine. Now, concentrated urine, okay? We got to get out of the drawing first. Sorry about that. Let's get to concentrated. It can be 4 times more concentrated than blood plasma. That's why I was pausing on my math there when I said it should be 5 times more dilute. It requires antidiuretic hormone, right here, okay, to increase the osmotic gradient. Right, oops, Control Z. There we go. Right, and there are 3 major solutes that are going to contribute to this. We will use sodium chloride and urea, and we're going to build and maintain a gradient, which will then cause a countercurrent flow in fluid through the descending and ascending limbs of the loop of Henle. You're like, what? It's okay. Hopefully I'll walk you through it. How does this work? We have to do something that's called countercurrent multiplication, where you have a progressively increasing osmotic gradient as a result of countercurrent flow of fluids. Symporters in the thick neck of the ascending limb allow for a buildup of sodium and chloride concentration in the renal medulla. Okay? Remember I said we're going to take advantage of the fact that cells are impermeable to water, so therefore we drive the want for water. That establishes the reabsorption of sodium and chloride as it becomes more and more concentrated, as well as cells in the collecting duct reabsorb both water and urea. Urea is the product of deamination for amino acids in metabolism. Urea recycling causes a buildup. Urea recycling. Okay, and that's going to create that countercurrent multiplication. You'll see a picture of it here. Everything I just told you is now just bringing in the one last piece, and that is, where's all the material going? The vasa recta is the countercurrent exchanger, meaning if materials are coming out of the tubule, what are they traveling into? They're traveling into the circulatory system temporarily. Okay, and you'll see how this happens. So here's the picture that's inside your textbook, and it's fairly confusing. And the first thing I would always like to remind you is this piece over here belongs over here on top, just like they did up here. In years past, they didn't do that. People were really confused. And notice what's happening that I showed you before. The osmolarity is decreasing, and because this is the juxtamedullary nephron, you actually have 1,200 millimoles of fluid here, and look over at the vasa recta, you can see that there is a slightly different, means exactly the same value here. But when you look at the values inside of the vasa recta, if you put it on top here, water can move out here, sodium and chloride can move out here, and we can recycle the water to dilute the sodium and chloride that's temporarily being brought up and then sent back out. Okay? The process of doing this, take a look at what's happening to here, the osmolarity inside the bloodstream, right? So you're slightly increasing the osmolarity here. And take a look at the osmolarity here at the proximal convoluted tubule is 300. By the time we finish using urea to concentrate the fluid of water inside the tubule, because the urea is outside here and the sodium chloride is outside here, so it exits the distal convoluted tubule as well as the collecting duct, you now have concentrated urine. That's why your urine gets so yellow. So we've done all this work. Hopefully it all makes sense. Why did we do it? Well, here is a summary of everything that actually happened. I think inside class I said nudge, nudge, hint, hint, wink, wink. Make sure you know pretty much who's being reabsorbed and secreted, where. But more importantly, I think I don't want you memorizing exactly how, where, when. That's a lot of useless information inside your head. Try and figure out, hey, hydrogen protons. Well, we know I need to regulate pH with those. You'll notice hydrogen protons only show up a couple places. Why is that? Okay. Bicarb. Bicarb is pretty important. Yes, no, maybe. Okay. So in lab, we did the urinary analysis. Some things we couldn't test for, but primarily it's the best indicator of what's going on with your kidneys, something that happened inside, 10 minutes ago inside your bladder, or maybe a half hour ago inside your blood, inside your blood that's been now filtered out and now stored inside your bladder. What you're looking at, these are two types of tests that look to make sure your kidneys are actually flowing properly, right? Okay. Blood now travels out. Listen to me. Urine now travels out through the ureters to travel from each side to one bladder, where peristaltic waves and hydrostatic pressure as well as gravity essentially move it. So what's going to happen is the bladder is going to fill and it compresses the opening to prevent backflow. So let's take a look here. Ureters travel down, urethral openings, pressure inside here, these rugae, actually close those openings to make sure that fluid stays in there. You actually have the involuntary sphincter here. This is when you're running to the bathroom and you can't hold it anymore. And you actually have the voluntary perineum muscles here. And this is for female, but you can understand the same things, generally speaking, happening for males as well. And the idea is this is going to travel out through the urethra for males. Females, what's going to happen is this is what you're pinching when this is letting go before you can actually sit down on the toilet. Okay. The bladder itself can hold about 800 mils. You can actually get it to hold much more than that, but everything else considered, there's no, unless you have a real need to hold that much more, whatever, who cares? Big fancy word for peeing is, of course, micturition. Okay, that's the discharge from the bladder. It's a combination of voluntary and involuntary. In early childhood, that's where we learn to initiate it and stop it voluntarily, right? Finally, the end of this chapter, okay, is stuff that you saw back in the reproductive systems. And I'm kind of hoping nobody makes this mistake inside lab that they did inside for the reproductive stuff. So remember, here's the bladder here. It's still smooth muscle, just like the uterus, but you'll notice it actually has a distensible nature to it, meaning this can stretch. You see all those folds and those rugae there, and that means this is the urethra here. This is the external urethral orifice. Male bladder, same general structure. Materials travel out, travel through the tube, and they then exit here. Okay, so we've covered a lot in these 3 sessions, right? And what I want to make sure you're paying attention to is, gee, that was easy. Urine comes out. Big fancy word, though, micturition. Hormonal regulation of target tissues inside the kidneys is really important. What changes glomerular filtration rates? Notice that was primarily neuronal and or mesangial cells with constitutive nitric oxide expression that is then temporarily shut off. Okay, we'll cover either this or 27 inside class on Wednesday. Whoops, that's probably too much information. Okay, be safe. Have a great night. Bye.